you had some changes in your career path here recently, and so tell us a little bit about yourself and what that looks like for you. And you are the 2025 Honeybee Princess this year, right?
Yes, I am the American Honey Princess for 2025 with the American Beekeeping Federation. I'm actually in North Dakota currently. This is my 12th state that I've been to. And based on kind of the things that I've experienced over this year and, all the wonderful things I've had, I've been switching over to starting my teaching credential starting in January of 2026. So I'll be handing over the title to the next girls for Queen and Princess in January. The very next day, I get to start my teaching credential over at the Concordia University of Irvine in life sciences for middle school and high school teaching. So I'm very excited about that. And yes, hi, Emilia Burnham.
Well, congratulations.
Thank you so much.
Thank you for your work this year
Thank you.
in the industry. So, Dr. Webb, you have an impressive bio. So thank you for being with us tonight. So
for having me.
you got your bachelor's at Jackson State, if I recall right, and then you had a kind of a big, I think, like a life change, right? You received a research fellowship from the National Oceanic and Atmospheric Association, right? And so you were studying marine invertebrates, which is that's a big old field, right? There's a lot of them in the ocean.
Oh yeah.
So anyway, that got your interest in beekeeping started, and you did an undergraduate research through the National Science Foundation at Michigan State, which if I understood this right, is a Kellogg's biological station, right? That's
Yeah,
is that
exactly.
a division of the university or?
Yeah, just like an offset of their campus.
That's right. So your research was in prairie restorations, and that's where you got your start in being interested in pollinators.
Yeah.
Right, butterflies and wasps and bees, honeybees.
Exactly.
Right, and that led you to a PhD in entomology from where you're at today, right?
Yep.
At CIBER. So, and you're there with Boris Baer, if I remember right.
I was, yeah, just recently graduated. So
yeah, good for you. Congratulations on that. What'd you do your thesis on?
well, you're going to hear all about that today, actually.
All right, that's what we were hoping. Thank you for that. Because as I remember, your thesis was on some work on drone, some sort of seminal fluid.
Yeah, we're going to touch on seminal fluid and how that interacts with Nosema and immunity and all that fun stuff.
Oh, that's great. Well, we're excited to hear from both of you tonight. It's sort of a forum, and you guys have coordinated some share time, I think, on this call or something like that. So we're glad to have you do whatever you feel like you need to do. To everyone who has stayed with us tonight, despite the challenges, we do record these, and these are added to our library in the WAS website, and which is changing soon in January, and we're excited about that. So more to follow in that area. And also we have a question and answer period. I think when we did our prep for this conference, we let you know that there is a Q&A at the end of this. So anyone who's on the call that has a question for either of these presenters, put them in the answers and questions and not in the chat. And that's where we'll go to answer them. So the time is yours. And thank you again to both of you for hanging in there with us.
Yeah, of course. So I'll just go ahead and share my screen. Oh, well, sharing is not turned on. So I sent a request for that.
It should be able to if you try again.
There we go. Okay, so hopefully everyone can see
Yes.
now. Awesome. Okay. So thank you, Scott, for the introduction. Again, my name is Jessica Webb, and I'm just going to share a little bit about some of my dissertation work over at CIBER. And if you're not aware, CIBER is at the University of California, Riverside, so Southern California. Again, I don't have to share this with you guys, you already know. The honeybees are experiencing an average of 55% losses, and this has been going on since around 2011, right? And this year was particularly bad for our honeybees. We've seen losses of more than 60%. And we can attribute this to quite a few things, right? So pesticide exposure, climate. But one of the major drivers that we know is causing these declines are pathogens and parasites. One parasite that I'm particularly interested in is Nosema ceranae. And so this is what Nosema looks like here. It's these just kind of ovular shaped parasites, nothing super significant looking. But these parasites are really important, and they've had quite a journey since their discovery. So I'm going to talk a little bit about their history. So in 1909, Nosema was first detected in Apis mellifera, not necessarily here in the US, but in Apis mellifera. And then in 1996, we see the discovery of Nosema ceranae. And this was described in Apis cerana. So to be clear, Nosema apis here and Nosema ceranae here are two completely different parasites, okay? So again, at this point, Nosema ceranae wasn't actually found in Apis mellifera yet. By 2005, that's when we finally see the appearance of Nosema ceranae in Apis mellifera. And at this point, the Apis mellifera, or our European honeybee, had both Nosema ceranae and Nosema apis. But this didn't last very long, because in 2011, we started to see less detections of Nosema apis. So Nosema ceranae had pretty much outcompeted Nosema apis. In 2020, we had some systematists, which are people who are responsible for categorizing and naming organisms. They decided that Nosema ceranae should actually be named Veramorpha ceranae. And of course, us bee scientists, we didn't really like that. We thought it was super confusing for us. We thought it was confusing for the beekeepers, for the general public. And so in 2024, we fought back and said, you guys can call it Veramorpha ceranae, but we're going to keep calling it Nosema ceranae. So till this day, we still call it Nosema ceranae, and that's what I will refer to it as in my talk. Nosema ceranae is found on pretty much every continent. The only place that it hasn't been found is Antarctica. It's most prevalent during spring and summer, and especially in areas with higher humidity. But of course, this is going to vary based on temperature. So in SoCal, we very often will see it in late winter, early spring. So what exactly is Nosema ceranae? It's very often miscategorized as like a virus or a bacteria, but what it actually is a microsporidian, which basically just means it's a really tiny kind of fungi spore. So it's really closely related to fungi. And it's an obligate microsporidian, which means it depends on its host to survive. And so in this case, the host are our honeybees. So again, it has this really prominent oval shape, and it's about 4 to 6 micrometers in size. So it's not going to be visible with the naked eye. It's typically spread through ingested spores, so via trophallaxis, which just means bees feeding each other. Grooming can also spread it, so grooming themselves, but also grooming each other. Or it can be spread through contaminated honey or nectar. And so this fungal spore or this microsporidian is typically gonna infect the midgut of the bees, but not always. So that's gonna come in handy a bit later. And the way that it infects its host is it has what's called a polar filament. So this is our spore. And on the inside of it, you kind of have this thing that looks like a slinky almost, but it's spring loaded, so it's pressurized, right? So once the spore is inside of your midgut, what happens is this spore basically shoots out, and it shoots this tube out into the cell wall of the midguts. And then it's just able to eject DNA into the cell and eventually asexually reproduce inside of the tissues before it shoots out of the tissues back into the midgut. So some common symptoms that we see with Nosema ceranae is dysentery. So these poop smears outside of the colony. It's not super common that we see it with Nosema ceranae, but it was a really big telltale sign with Nosema apis. The bees do experience nutritional deficiencies when they're infected with Nosema ceranae. And ultimately, those nutritional deficiencies will cause your bees to be more lethargic, and you'll see reduced foraging efficiency. So you're going to see reduced foragers at your colony entrance, and you might see a reduction in your honey yield. But ultimately, Nosema ceranae it's not super virulent on its own. Like, you're not going to see generally colony collapses due to Nosema ceranae. Nosema is often characterized as a secondary parasite, which can mean a couple things. Because it does suppress the immune system, it does make it harder for your bees to fight other infections or other parasites or other pathogens. But it also means that if your bees do have other parasites or pathogens, then it will make it easier for them to get Nosema. So that's kind of what we mean by a secondary parasite. That doesn't mean it's less dangerous. Just on its own, it's not going to be super virulent. How can we detect Nosema ceranae? So that's a bit more difficult. As scientists, we have molecular techniques that we can use. One, for example, being qPCR. And with qPCR, we just measure the amount of DNA, of Nosema DNA that we find inside of the honeybee guts. But to do qPCR, that's several $1,000s and lots of fancy machinery that a beekeeper should never have or never ever need. You can also use a microscope, and a good microscope that gives you the strength that you need maybe cost around $200, right? So it's a decent investment because, you know, microscopes last forever. There's also bee disease diagnostic centers in the US. So USDA has a really good center for it. University of Maryland also has a disease diagnostic center. So you can just pack up some of your dead bees, ship them off to the lab, and they can tell you all types of viruses and bacteria and other things that you might have in your bees that you can't necessarily see with the naked eye. The issue with Nosema is that even if you are able to detect it, there's not a true established treatment threshold. We have one for Nosema apis, and I think people have kind of just assumed that that also works for Nosema ceranae, but to be honest, we haven't really done the work needed to establish a treatment threshold like we see with things like Varroa. Like that's really well established, not so much in Nosema ceranae. For Nosema ceranae, there are treatments available though, but we'll loop back around to that at the end. So earlier on, I did specify that Nosema is a gut parasite, typically, but there are other ways that it can be spread. And one of those ways was actually discovered by complete accident. So the story goes, there was some grad student in the lab looking under the microscope, and this is the slide they were looking at. And if you can't tell, what those squiggly lines are is actually sperm, right? So this grad student was looking at sperm under the microscope, and lo and behold, what they actually found were these Nosema spores, right? So this is the first time that Nosema spores have been found in the honeybee's ejaculate. So for the purpose of this talk, ejaculates just means the sperm plus the seminal fluid. And what seminal fluid is, it is the liquid that, you know, protects the sperm and helps it, makes it way to the reproductive tracts of the queens. So this is really cool, but it's not uncommon for gut parasites to be found in the reproductive fluids, especially for small organisms like bees, where the reproductive organs aren't, they're really close to where the bees are pooping as well, right? So their anus is really close to their reproductive organs. So it's not uncommon for there to be contamination for something like this. But thankfully, my lab is really good at artificial insemination. So we can do these really controlled experiments to see if what we're seeing is actually a sexually transmitted disease or if it's just contamination. So one way they were able to do this, they started with our honeybee queens. And then they artificially inseminated Nosema spores into the reproductive tract of these queens. Just the Nosema spores, nothing else. And so what they found is that over time, in the spermatheca, the ovary, and the gut, they seen more Nosema spores than what they put in, right? And this was true for more than 55% of the queens that they inoculated. So to us, that indicates that yes, Nosema can actually be a sexually transmitted disease from the drones to the queens. So they then wanted to know, is there some type of defense system that the drones possess that would prevent the queens from becoming infected with these Nosema spores? So to do that, they again did artificial insemination with the Nosema spores, but they also added in the seminal fluid this time. And so, yes, they seen that there was a significant decrease in the amount of queens that were being infected once you add the seminal fluid in with the Nosema spores. So again, yes, that indicates that there is something in that seminal fluid that's actually protecting the queens or that's killing these spores. So they dug a little further into this. And this time they removed the queen from the equation. So they just had their Nosema spores and their seminal fluid. And so what they did was they exposed the Nosema spores to the seminal fluid to see how many of the Nosema spores would actually survive the exposure. And so on the left, you have your seminal fluid, and on the right, you have your control. And in this control, it's basically just sugar and salt water. So when the Nosema spores were exposed to this control, about 70% of them were perfectly fine, perfectly viable and alive. But then once exposed to the seminal fluid, you only see about 5% of these spores are able to survive. So again, there's something in this seminal fluid that's actually killing these Nosema spores. So they broke this down a little further. The seminal fluid can be divided into two parts, the non-protein fraction and the protein fraction. So they started by investigating the protein fraction. And they found that there's over 250 different proteins in the seminal fluid, and all of them have their own separate functions. But one that really stood out was called chitinase. And so what chitinase is, its whole job is to basically degrade chitin or to eat chitin. And that's super important because we know our Nosema spores are actually encased in a chitin cell wall, right? So the thing that protects them is made of chitin, and this chitinase is just going to eat right through it. So that makes sense why the protein fraction would be able to decrease the amount of alive spores that we were seeing. So then we loop back to our non-protein fraction. And that's a really vague term, but we know that that non-protein fraction contains what is called metabolites. So again, they dug further into this, and they found that the metabolites, or that the non-protein fraction contained about 54 different metabolites. And there were some that were super interesting, that were really standout metabolites. And that was your benzoic acid, your citric acid, and your oxalic acid. And that's because we know that these three metabolites have been used previously as antifungals, right? So in antifungal creams or things like that. So this is where my research comes in. I wanted to know, can we utilize these metabolites to mitigate the intensity of Nosema ceranae infections? So just to reiterate, we have these antifungal metabolites that were found in the seminal fluid specifically. So again, benzoic acid, oxalic acid, and citric acid. And we have our microsporidian, which is super closely related to fungi. And we find these typically in the guts. So I wanted to know if we use these metabolites as an oral treatment to attempt to reduce Nosema ceranae gut infections, right? So the metabolites plus the spores should equal dead spores was kind of my thought process. So in order to test this, I had to individually infect honeybees with Nosema spores. So you can see our honeybee here being hand-fed by our pipette with the Nosema spores in them. And after 6 days, I treated them with a mix of the metabolites, and I just mixed them into sugar water feeders. So you can see an example of what they would kind of look like in their cages. After 12, 15, and 18 days, I then dissected the midguts of all the individual bees, macerated them, and then we used what's called a hemocytometer to then count the amount of individual spores inside of all these bees. And what did we find? So we found that we have that all three metabolites significantly reduced infection intensity compared to our positive control here, right? So our positive control were the bees that did not receive any treatment, and they had about 400,000 spores in their midguts. Whereas with all three of our acids, we see that it's pretty close to zero. We also looked at infection prevalence. So infection prevalence is the amount of bees that had any infection. So if they had one single spore, we counted it as prevalent for infection. The bees that did not receive any treatment had about 60% that were infected. But then we go to our treated bees, we see that this drops significantly, right? So only about 15% of the bees that were treated had any sign of infection at all. We then wanted to see if there were any negative effects of using these metabolites on the bees. So we looked at survival. So how long were the bees able to survive during treatment when compared to our positive control? And this graph is a little hard to read, but it basically means that we didn't see any difference in the survivability of these bees that were receiving treatment compared to those that weren't receiving treatment, which is actually a really good thing. It means, you know, there's no negative effects of the acids on the bees. We also looked at mortality rate, which is kind of goes hand in hand with survival. So here's our positive control. We see about 90% of those bees are going to die when they don't receive treatment, but are infected. And these are our three acids again. So while there was no significant difference in oxalic acid as far as mortality went, we also didn't see an increase. So again, no negative effects. But here for benzoic acid and citric acid, we see a significantly reduced amount of mortality, right? So around 40% compared to 90%. So about half as many bees are going to die when receiving these treatments. So from that initial experiment, we can kind of conclude that, yes, there are metabolites in the seminal fluid that show antimicrobial or anti-nosema properties. And when the metabolites were added to the infected bees' diet, they were able to reduce infection prevalence and intensity in the gut. And there was no increased mortality or decreased survival, which indicates that there's no immediate negative impacts of the metabolites. So these metabolites do have the potential to be used as a medication at the colony level, or at least that's what we concluded from our initial experiment. So I did test that, of course. For my second objective, I wanted to see, again, could these metabolites be used under field conditions? So at the colony level, can we use these as a treatment for nosema? So I promised I would talk about some of the current nosema medications that you could possibly use or that exist. So I think the big one, the main one that people know is fumagillin B. You might also hear it referred to as Fumadil B. And what it is a powder that's derived from Aspergillus fumigatus. And what we know about Aspergillus is that it is an antibiotic, which means it's actually meant to kill bacteria, not microsporidians, which is what nosema is. So it's interesting that we use this in the first place when it's not really what its intended use is for. It was originally used to treat Nosema apis, but again, this was recommended before we had actually done the research to see if it was equally as effective for Nosema ceranae. And today what we kind of see is that the efficiency is inconsistent. So sometimes it works, sometimes it doesn't work. We also know that when people use fumagillin, the Nosema spores tend to build resistance to the medication relatively quickly. Yeah, so I don't actually think it's FDA approved in the United States at the moment. I do think maybe in Europe and Canada it was FDA. Well, it was regulated for a while, but I'm not sure if that's still the case actually. There are other things available or that people use. So there's probiotics like Protexin. And yes, this is a picture of a dog on the bottle. That's because, again, it's not really meant to be used in bees, but people certainly do. There's vitamins available. So, VitaFeed, Gold, and you also have things like Nosevit, which if you look on the bottle, it's just kind of labeled as an intestinal cleansing. But the thing about these available medications is that none of them are actually labeled or meant to kill Nosema spores. They're kind of targeted or like marketed as things that kind of give your bees an immunity boost. But again, they're not really meant to target or kill these Nosema spores. So, if there was a medication that had that ability, that'd be pretty cool. So that's why I really wanted to test these metabolites that worked in lab in the field to see, is there a possibility that we can use these metabolites under field conditions? So in order to test that, I started by doing what's called equalizing colonies. So we basically kind of played Tetris with our colonies. We wanted each colony to contain 5 frames of capped brood, 2 frames of nectar or honey, and 3 empty frames so that the queen had space to continue growing. And we wanted to make sure that there was a population of 8 frames. So we wanted at least 8 out of the 10 frames to just be full of adult worker bees. And so after we let the colonies establish for a couple weeks, I then created this method where I could infect the colonies all at once. And so to do this, I basically sprayed 125 million Nosema spores on the inner 8 frames of the colonies. After 14 days, we divided these colonies into 2 groups. One group was treated, and then the other group did not receive treatment. And so for the treatment, we did very similarly to what we did in lab. We mixed our metabolites into a honey water mixture. And again, we used the exact same dose that we used in lab. We then place that metabolite mixture in mason jars in front of the colonies. And so we used this really cool bright red dye that kind of made the metabolites look like Kool-Aid. It was very tempting to drink, but I promise I never did. But we used this dye because we wanted to be able to track which of the bees had actually consumed the mixture. So every 2 days, we went to the colonies and we collected foragers at the entrance. And again, we did exactly what we did in lab. We dissected them and then we counted the amount of spores they had in their guts. And so you can see here on the left, you have your normal midgut that, had not consumed any of the metabolites, and our bright red ones on the right because they had consumed the mixture. We also performed colony evaluations every 2 weeks because we wanted to know if there were any negative effects on the colony. So we wanted to make sure that the queen was still laying eggs and that the larvae and the brood was doing fine, and that the bees were still bringing in pollen and nectar and had plenty of capped honey, and their population was still doing relatively well. Okay, so before I show you guys the results, I want to make sure that they make sense because there are basically two different things going on. So the first thing we have is our pre-treatment. So we wanted to make sure that the infection method that we used actually worked. So we call that the pre-treatment phase. That's from day 0 to 14. And again, we just wanted to measure to make sure that there was actually going to be an infection before we started treating. So that phase will be indicated in black. And then we have our post-treatment phase, and that's from day 16 to 28. And then I measured again 1 month later at day 56. In this part of the results, the colonies, again, are separated by treatment. So in brown, you'll have the colonies that were treated, and in yellow, you have the colonies that were not treated. So again, this is kind of a lot to look at, but what's important to take from here is that there was no significant difference in the colony metrics that we took. So population, nectar, eggs, pollen, capped honey, and capped brood. There's no significant difference between treatment, which again is really good because that means there's no negative effects of these metabolites at the colony level. The only metric that we've seen a significant difference was in larvae, and we're still not really sure why that was happening, but we don't think it was an effect of the metabolites themselves because we see this difference before we even start the treatment, but by the time we get to the end of the treatment, we see no significant difference. So we then looked at prevalence, which again is just the amount of bees that actually did have an infection. And so in the pre-treatment, we see that there was an increase in prevalence, which is good because that means the infection method that we used actually worked. So the bees were infected before we started treating them. And when we started treating them, we see that both our treated and our not treated bees did decrease in prevalence, so the amount of bees that were infected. But we see that the amount in our treated groups is actually greater than those that were not treated, which is good because it means that there were less infected bees than those that were treated. And so we did the same thing for infection intensity. And so intensity is the amount of spores inside of the bee's guts. So again, in the pre-treatment, we've seen an increase, which again is good. And in our post-treatment, we see a decrease, again, in both our treated and our not treated groups. And so we see the same pattern here where our treated bees are significantly lower or have significantly less spores by the time we get to the end of the treatment period, which indicates that the treatment isn't effective. But we were really interested here at this day 28, so we just wanted to get a closer look because this is about 2 weeks after treatment. So we compared this day 28 to the largest peak infection, and we see that the bees that were not treated, there's actually no significant difference from when they had the most amount of infection compared to those that were treated. So again, we're seeing that this treatment actually works relatively well. So just to kind of summarize that particular objective, we did, create a new way to infect colonies with Nosema, but thankfully we were actually able to decrease them using the medication that we found or that we created. And we didn't see any negative effects of the treatment on our colony metrics. Yeah, and there was a greater reduction in spore prevalence and intensity in our colony treatments than in our not treated colonies in the long term. So at this point, I was super confident in my medication, right? I thought I had found my $1 million idea, and I was super excited to share this with our local beekeeper. So this is me giving a talk at one of our local beekeeping clubs. And very often when I would give my talks, some of the feedback that I would get would be, Jess, this is really cool, but Southern California beekeepers don't get Nosema. And so this was really confusing to me because I consider myself a Southern California beekeeper, and I know we have plenty of Nosema in our colonies at least. So the scientist in me, again, was super curious and wanted to know what was going on. So I did the only thing I know how. And so I did an experiment. I got the data. I ran the numbers because I really wanted to know. So that was my next objective here. I did a survey. So basically I asked a bunch of beekeepers a lot of questions. And the first question that I asked was, do you have Nosema in your colonies? And let me tell you guys, I was very humbled very quickly because almost 99% of the beekeepers said, no, we don't actually have Nosema in our colonies. So again, very humbled, very sad also. So then I asked this next question, which was, would you be interested in a new Nosema medication if we can get this to work? And based on our last question, you guys shouldn't be surprised on what the answer was here. 75% of beekeepers said, no, we would not actually, we don't need this medication for our colonies. And so again, I felt like I spent a lot of time creating this medication for, Southern California beekeepers. And it turns out maybe they don't need it specifically. But I wanted to dig more into this. So we asked quite a few questions on our survey. We wanted to know where these beekeepers have their colonies, where they're getting them bees from, what pathogens and parasites they do think they have, and what's the management style of the beekeepers? So what type of medications are they using, and how often are they checking their colonies? So again, we just used Qualtrics to create a questionnaire, and we had about 121 participants. So it's a pretty big survey. And again, all these beekeepers are from Southern California. So I'm not going to go through all the results, just some of the more relevant ones. Another question we asked was what parasites, pests, or pathogens they thought they had in their colonies. And of course, some of our bigger ones were varroa mite, hive beetle, and wax moth, poor Nosema all the way down here. Pretty irrelevant, it seems, maybe. But another interesting question we asked the beekeepers where did they source their colonies? And so we've seen about 35% of them were keeping managed or commercial stock. But another 29% was actually keeping feral stock colonies in their apiary. And then a group here, which was 35%, were keeping both managed or commercial stock and feral stock. So we were able to divide the data to see if that was possibly playing a role in the results of this survey. So we've seen that with some of these diseases, with some of these pests, there were actually differences in how many were being reported based on where these beekeepers were sourcing their colonies. So varroa mite, for example, we've seen that it was reported significantly more in our commercial or our managed stock colonies. Now, I won't go too far into this because I think our next speaker has probably quite a lot to say about it. But we can summarize a few things from this survey. We found that a large portion of beekeepers in Southern California are keeping what we call survivor stock or our feral stock. Feral bees and managed bees could potentially have different parasite and pathogen loads, but of course, further studies would need to determine if there was any effective genotype on parasite and pathogen loads. So just to conclude my portion, in our objective 1, we found that bees do have these cool immune defenses in their seminal fluid that have antifungal activity against Nosema ceranae infections in the gut. In our objective 2, we found that while these metabolites have the potential to be used as a Nosema treatment, the bees were actually able to reduce the spore loads in the gut on their own. So, maybe the bees have, hope for themselves and maybe they don't need a medication or maybe they do. I don't know. Our objective 3, we found that local beekeepers don't necessarily see symptoms of Nosema in their colonies. So the question then becomes, but why? And so with that, I'll save all questions until the end, and we'll let Emilia talk because I think she might actually have some answers for that.
Maybe so. Maybe so.
Okay.
All right. Awesome. So you'll see a change in colors here. So I'm a very pinky girl. I love hearing Jess talk. She was my mentor actually when I first started out in the lab. So I know it's very nice. So I'll start off then bringing in talking about that honeybees were bred for really strong honey production because of course we want our bees to be really good honey producers. But of course, as in the survey, also people want to have decreased defensiveness in their bees. So continuing on, then hold on, could we go next slide? I'm realizing that Jess is still sharing the slides. Do we want to switch that over? Or do you want to control them? What do you think? Okay, cool.
Switch it. That makes sense.
Yeah. So thank you. So then, yeah, let's switch that. Which means I need to get out of full screen here. Bear with me. Bear with me on this one. Come on. It's been a day. Why is this happening? I just want to present. I apologize.
Are you having issues with sharing?
I'm just trying to find it on the site. That is my only issue, and I don't understand what's the matter.
Jess, you could put yours back up if you wanted.
Yeah, sorry. Yeah, like literally I don't know what's wrong with this on my side.
Okay.
Sorry. Thank you. Okay. I guess, yeah, we'll just keep going from here then. Okay. So then, since we know that honeybees are bred for strong honey production and decreased defensiveness, that means that honeybees are also going to have compromised ability to defend, possibly. Next slide. So the solution for that is exploring new genetics. And then if we continue on here, I looked at 2 genotypes of honeybees that were also investigated in the survey. We're looking at the California honeybee, which are the more aggressive Africanized honeybee, but we call them California honeybees in our lab, and then also looking at the more European commercial honeybees. So my primary objective here was to compare the immune responses of the survivor and the managed genotypes, these California and commercial genotypes, and challenge them with the pathogen Nosema ceranae that we're all talking about today. So next slide here talks about my hypothesis. So I suggested that the Californian honeybees would be more resistant to Nosema ceranae than the commercial honeybee, and that would be because of the mixed genetic diversity there. So I believed that then the Californian honeybees would have a stronger survivability. They would be able to tolerate pests and pathogens way better than the commercial genotype. But we'll see what that happens and looks like. And I thought that they would have better immune systems. So my prediction here was that when I fed honeybees with a heat-killed version of the Nosema spores, that the spore levels would be decreased and that the vitellogenin, juvenile hormone, and chitinase would also be decreased. Then going on, then to test this, I used the Manage and Survivor honeybees, the commercial and Californian genotypes at UC Riverside and Nosema ceranae. So that's Nosema there. Next slide. Then I studied innate immune responses. So there's two different types of immunity, but I'm focusing on just innate for today. And that really focuses on hemocytes. Those are basically the blood cells in the honeybees' bodies, and those are responsible for the cellular responses. And that also includes antimicrobial peptides, signaling pathways, and these hemocytes exist in the honeybees' hemolymph, which is their insect blood-like substance. So going on, my prediction was that in the honeybees' innate responses, when that innate immune system would be activated, it would activate the signaling pathways, and it also increase the hemocyte activity. That was my main prediction there. And how did I measure these responses? With biomarkers. So I looked at three main biomarkers. I looked at vitellogenin, juvenile hormone, and chitinase. So Webb did a really great job talking about chitinase, and then I'll talk to you more about vitellogenin and juvenile hormone. So vitellogenin is it's a lipid, and it transports the pathogen fragments into the egg. It's very involved with the egg that the queen lays, and it helps reduce susceptibility to pathogens and parasites. And it's been found that the nurse bees have higher vitellogenin than the forager bees do. Then juvenile hormone controls the age-related transition from bees being nurse bees to foraging adults. So as the bees get older, they increase in juvenile hormone, whereas when the bees get older, they decrease in vitellogenin. So it has this kind of inverse relationship, which is pretty interesting to me. And it has been found that juvenile hormone can help defend against insecticides and protect the midgut cells. And then chitinase, since Webb kind of already talked about it, I won't say more, but I'll just say that enzymes, they are the enzymes that degrade the chitin that are found in the exoskeleton's cuticle. And the chitinases, like she said, have been found in the honeybee seminal fluid and have been predicted to kill Nosema apis. So those are my three main biomarkers, and now we can talk about the methods. So in here, I have the treatments, and I used adult worker bees that were less than 12 hours old because I didn't want them to be able to be infected by Nosema. So I've got my no manipulation treatment. So these were the bees that I just took straight off of the honeybee frame and took them right off the cage and put them into the cage. So that's my main control group. My next control group was the sugar water treatment. The sugar water treatment, I fed bees with 2 microliters of sugar water before I put them into the cage. So that's the only difference between the no manipulation and the sugar water treatment. So positive control. Then I have my dead spores treatment. The dead spores treatment were fed 2 microliters of dead heat-killed spores, and they were put into the cage. That was one of my experimental groups. And the last one was the live spores. They were fed 2 microliters of live spores before they were put into the cage. So another experimental treatment. So 4 treatment groups. And with these 4 treatment groups, I put them each into separate cages. Each treatment had 3 replication cages, so 12 cages in total. There was 30 bees in each cage, and they were also given 1-to-1 sugar water, and they also had water feeding tubes. So that's where they stayed for the entirety of the experiment until I pulled them out. Then in each cage, I pulled out 5 bees each day to identify how identify how intense their infection was and how the biomarkers were. So I pulled out their blood their hemolymph for each day on days 6, 12, and 15 days post-infection. And then their midguts I pulled out on those same days to analyze both of those aspects. Then on here, with the hemolymph, I investigated for juvenile hormone, vitellogenin, and chitinase. And in the midguts, I did spore counts on each of those days, and then that told me about the infection intensity. So now I can tell you about how I heat killed the spores. So you can use an autoclave, but at our university, the autoclave was not always super effective. Well, because, it's always broken. So we used a pressure cooker. So my friend Mark Swenson helped me figure out that we could use a pressure cooker with a ramekin with a little beaker inside to kill the spores, and they were very effective. So we knew that this was an effective method of killing spores for my experiment. So I gotta thank him for that one. Then I used tweezers here to poke a hole into the segments between the second abdominal segment. I went in there with a little needle to pull out their blood. So I did that to remove the blood, and then I went through the stinger to remove the midguts. So that's just the methodology there, and the midguts right in there. As seen by this pink square, that is the midgut that I pulled out. And then I did 3 different experiments. For the biomarkers, I did a vitellogenin assay to see how much vitellogenin there was in their blood. I also did a chitinase assay and then a juvenile hormone assay with my 2 mentors here, Dr. Naoki Yamanaka and Dr. His name is escaping me. Next slide. So then we go into results and analyses. So to begin with, I was looking at different months of my experiments, but we're focusing on August and November here. So what was found was that in the summertime, the bees were more likely to have infections in the commercial genotypes in the summertime, and then obviously the survivor bees, these California bees, they had higher infections in the survivor group in the November. So there's very strong differences between the August and the November treatments. Then we move on to see the vitellogenin treatment, the vitellogenin tests, the live spores and the dead spores. The vitellogenin was not a very strong indicator of how the bees would respond. So there really was not much difference between the 2 groups. There's nothing significant here. But where there is significance was in the genotype on the next slide with just the no manipulation bees. So with the no manipulation, we do see a difference in genotype of how they respond between 6, 12, and 15 days. So we know that, yes, there is a difference there, and that's something that we can look towards more in future experiments. Next slide there tells us about juvenile hormone. That was the strongest indicator of the experiment and how bees would respond. So between lab spores and dead spores, we can see that, yes, these Californian more aggressive Africanized bees, they do have increased levels of the juvenile hormone versus the commercial bees that have really low juvenile hormone. And that is something that you'll see looks different than the next slide, which will show the no manipulation in the sugar water, whereas, yeah, that looks way different. The commercial bees had much higher levels. So that's something there that tells us, this might be something we can look into and might be an indicator of how honeybees respond to Nosema ceranae. Then we have the chitinase experiment. The chitinase experiment between live spores and dead spores, there's good indicator of significance in the genotype and the day. When you look at dead spores, so we know that this was effective. The chitinase, there's something to be told when they're infected with Nosema ceranae. And then the next slide tells, really with no manipulation, there's not much significance, not much to look for in here. So then I look at how honeybees respond in the mortality. In their mortality, we see that the managed in sugar water and managed in live spores groups were the most significant, being that when these commercial bees were infected with Nosema ceranae, they were 1.8 times more likely to die versus the managed and no manipulation bees that, they did not die as likely. So they were able to survive longer than, when you see on the next slide, these survivor bees, the Californian bees, they died really, really, really quickly, up to 5.7 times more likely to die versus these managed bees. And this was very significant, which tells us that, yes, these Californian bees can't really survive longer than the managed bees do. So that is something also to be really on the prowl for, like, this is kind of interesting. Then we have the discussion. So I can tell you guys then that expectations are not always the reality. So these Californian survivor bees did have much higher, I thought they would have much higher resistance and tolerance to Nosema ceranae than the commercial bees. But the reality is that it's quite unexpected of what could happen. The results are very unexpected. Therefore, the survivor bees, these Californian bees, did have resistance without tolerance mechanisms. So they were able to resist infection pretty well, but they died really quickly because of that. And then the commercial managed bees had tolerance without resistance. So they were able to live a longer time than these Californian bees, but they had really high infections. So it's kind of like that interesting thing to play with there and look at. Then we have more information about possibly that the ability to tolerate the infection comes from the Western European, Middle Eastern, or the Eastern European genetic lineages, and possibly not the Africanized lineage. So that's just something you really want to look for maybe in the genetic side of things. I'm not going to be looking at that, but who knows, that might be something to look at and be interesting. Then the hemolymph data tells us a lot of interesting information that possibly the day 6 to 12 day period of the Nosema ceranae infection, this could be important for understanding how honeybees respond immune-wise. This is really important in the day 6 and 12 because this is when the bee's body is recognizing that the infection has been building up and it begins to fight it off internally. So day 6 and 12, it recognizes the infection. You've got that really high infection period, and then it starts to fight it off. So day 6 and 12 might be really important. Who knows? Next slide. So juvenile hormone, we really saw that the dead and live spores did really induce the production of juvenile hormone in the Californian genotype versus the commercial genotype. Then we see that chitinase, we see that exposing bees to live spores, it also induced the chitinase production in both of the genotypes. Hiccup. And then with vitellogenin, obviously there's not a lot to say on this because the data, was not very significant, but we do know that the no manipulation treatment had significant differences between the two genotypes. So we know that there's a difference in vitellogenin just genotype wise, but then the California genotype tended to increase across those 3 days in the experimental groups and the sugar water treatment. So we see that there's some interesting pattern to continue looking at in this Californian genotype. So to conclude, future directions, if I were to continue this, I would enjoy doing some field trials to see how this would look. I would love to see how the primary generation looks at picking up the spores and seeing how they would react to that. And maybe you could use this as a vaccine. That'd be really interesting to see how the future generations would react to that. And I'd rather use bigger sample sizes next time and more replicates for really good, deep, stronger analyses. Those are the future directions. And just in conclusion, I have here my research question again. So how did the immune responses of Californian honeybees and commercial genotypes of the honeybees react when they were challenged with a pathogen such as Nosema ceranae? So in here, we have a little chart I made. So for mortality with these Californian Africanized honeybees, they died. They did not survive very well at all. And then the commercial bees, they were able to last longer. Hooray. The Californian honeybees with infection intensity, they could survive it. That's great. But commercial bees with infection intensity, they were sick. So you can kind of see this inverse pattern again. Then in the next slide here, with the biomarkers, I had predicted that these all of the quantities would actually go down, but they had really interesting results. So juvenile hormone was very much increased in the Californian honeybees, but not as increased as the commercial bees. And live spores treatment told us that the most. The Californian honeybees with vitellogenin did decrease like I expected, and it decreased more than the commercial bees, and no manipulation told us that. Then with chitinase, the Californian honeybees had an increase versus the commercial bees, which had less of an increase. So, unexpected results create some interesting stories to tell. So I was happy to tell you guys all about that today. And I believe we have some great funding sources to share. Jess, do you want to talk about that?
No, I mean, mostly just a thank you to NSF and USDA for funding the research that we've done in the lab. And big thanks to CIBER, the Center for Integrative Bee Research, for, supporting our research. And I think now we can actually take questions if there are any.
Yeah, thank you, both of you. That was great. Very interesting. So thank you. So we do have some questions, some in the chat and some in the Q&As. But before we head down that road, just let me pose sort of a question to the both of you and see what sort of feedback you have for us on this. So this has been really fun to listen to and interesting. So let's say to the average beekeeper, right, the hobbyist beekeeper, who may or may not live in California, or may not live in Arizona, or may not live where I live in Montana. What would be the takeaway from this that you could offer them as hobbyist beekeepers with 2, 3, or 4 hives in their backyard and worry about Nosema as part of their as part of their bee care? What would be the sort of big picture takeaway?
You want me to go? Or
Yeah, you could start.
So I think for me, I think overall what we learned is that the bees themselves are not completely defenseless. They're not completely hopeless, right? We've learned that the bees actually do have a lot of immune responses that they can, throw at the Nosema if they are infected. Now, of course, that's not always 100% effective. And as beekeepers, we should help our colonies if they are infected with Nosema. Now, the level that you are going to help your colonies is going to depend on quite a few things. Again, location, so where you live. Again, here it's not a huge problem, but that does not mean that it's not going to be a problem in, colder, wetter climates for sure, right? Or if you order packages, we know they're more likely to have Nosema than, already established colonies. So for me, you do need to take action if you know your colonies have Nosema infections, but it's going to be important that you are actually diagnosing your colonies with Nosema and not assuming, right?
Right.
Because what we're seeing with medications like fumagillin is that these Nosema spores are building resistance to the medication like really, really fast. And not having a proper diagnosis is not, it's gonna make that worse, right? So first step, diagnosis. Second step is kind of up to you how you choose to treat it. I personally don't have a recommendation on what medication or what action you should choose to take, right? Because again, there are so few treatments that are around for Nosema, and none of them are actually, regulated or federally approved. So I think anything you can do to make your colonies comfortable during that time. So if that is using vitamins or using probiotics, or using special pollens, or make sure that you increase the amount that you're feeding your colony so that they don't have to go out and forage. So anything you can do to kind of give your bees a boost, I think is the best thing you can do.
Yeah, you answered my question exactly, Dr. Webb, and that is it's different depending on where you live, right? I mean, and and how you go about it. But the other thing is that you really don't know. So you have poopy hives, right? So
Which again
in
is
Montana,
not
what's that?
yeah.
What's that?
I was saying, which again is it's not the most accurate way to tell if
Right.
your colonies have nosema.
Yeah, that I guess that was my point. You may have poopy hives. I have poopy hives in spring here in Montana, which I have a microscope, a little lab in my house, and I test for Nosema. I don't find it very often. It's a form of dysentery that has nothing to do with the Nosema infection. It's just diet and health. So is that kind of what you're saying? Is keep them healthy?
Yep. Make sure you're treating for other parasites and pathogens, and if you can, because again, it is one of those secondary parasites.
Mhm.
So, having nosema is going to make those parasites worse and vice versa.
Right.
And even with diagnostic tools, it's really weird because we know that Nosema ceranae has a different growth curve than Nosema apis. So with Nosema apis, we see a pretty exponential growth, and then at some point your bees just kind of, are super sick and they might die. But with Nosema ceranae, we actually see a more curved growth pattern, which makes it really hard to know when you should actually, check your colonies, like when the peak would be versus when they would kind of like mellow out, right? So again, that makes it's a really hard parasite to diagnose. So it's one of those things that you really need to stay on top of that.
Yeah, good comment. So, and another quick question would be, uh, so the best treatment is preventative, in this case. So keep them healthy, feed them some of the stuff that you guys recommend that you saw good outcomes with, right? Probiotics, vitamins, keep them healthy. And don't all of a sudden run to the fumidale thing. So let's say that for some reason that you see it, what do you do? So you see a poopy hive, what would be your recommendation what would be your next step? Say you've done everything you can, but you see these poopy entrances and hives dying with poop inside on the frames and stuff like that. What would you do?
I mean, in my first instance, if I saw this, I mean, literally the first thing I'd do would be try to pull bees from the hive foragers to investigate and see if they had a nosema, like you were talking about with, diagnosing on the microscope to see if they had nosema. Obviously, that's the first thing that I'd want to do. But at that point, then if you know, then yeah, like literally like Jess said, I think it is up to you what you do with that. You could take measures based on the research that you see and try out different things, because the worst thing that could happen is that your colony dies and you find out, okay, that's what happened. So you could try things that you read about, the work that Jess has done or anything that I've done. You can give it a shot, and you know it's really up to you. That's a tough call to make, I think.
So do you consider fumidale to be a prophylaxis in these cases or not?
There's a lot of information on fumigillin that's still coming out, I think, about, people are saying that fumigillin has like decreased the honeybee's immunity, and then there's some information that's conflicting that. So I think there's a lot of information to still kind of be discovered about how fumigillin is working, because obviously fumigillin was really working great for Nosema apis, but for Nosema ceranae, I think because it's a different species, maybe there's a new approach that needs to be taken.
Yeah, great. All right. So let's go to some of the Q&A, and anybody else here in the research field before we go to the Q&A that has any sort of feedback or questions for these two wonderful scientists. Okay. All right. So we have in the Q&A, you two, we have a question. Does the treatment for Nosema first reduce the issue, but ends up causing an ultimate increase higher level than when treated? Good question.
Which
So does it
treatment?
have a ricochet effect, right? A boomerang?
If the question is specifically about the treatment that I presented, like my research,
Uh-huh.
I think it is. Because it was at 825. It had to been you. Yeah.
okay. We don't know. That was something that I was really interested in looking at. Um, I wish that I knew. It's really hard to keep bees alive in a lab long term. And that would have been the best shot that we had looking at it. So after 18 days, the bees just kind of die off anyway.
Right.
So again, we know with like fumagillin that we see this resistant effect where when you treat, it does die off, but then the spores that don't die become resistant to the treatment. So if you try to treat again in 6 months, for example, those spores aren't going to die because they've been exposed. They know what you're throwing at them, and so the spores that they are reproducing aren't going to be affected by that fumagillin, right? And so it's very well possible that, my treatment could have that same problem, but we don't think it would have, simply because with my treatment, what we were using were 3 different active ingredients. So that would have indicated that the Nosema spores would have had, basically 3 different weapons that they would need to have to fight against, instead of one with, typical treatments like fumagillin, for example. So I
Yeah.
don't know. I hypothesized that that would not be the case.
Yeah. Good answer. All right. It said, there's another question here. Is it true that NC took over in Apis? So do you know the answer to that?
Emilia?
Yeah, I mean, based on Jessica's timeline, yes, Nosema ceranae has really been more prevalent now, more than Nosema apis. So Nosema apis was much earlier in the 1900s. I didn't mean to sound that old about it, but yes, so it was more prevalent then versus now, where really Nosema ceranae has taken over mostly. But
I
Okay.
don't think we've seen Nosema apis in the US again since maybe 2012, 2013. In other countries, I think they still maybe coexist, but as far as I know, not in the US. Nosema apis is gone.
yeah, I mean, I was actually just talking to Scott Opicultural over at the USDA, and they're doing a Nosema project right now to, figure out different detecting like diagnostic things. And from what the bees they're seeing over here in North Dakota, that Nosema apis is not in any of these colonies, and Nosema ceranae is about 80% prevalent in these colonies. So Nosema apis is not even there anymore. So
Interesting.
yeah.
That's good to know.
Mm-hmm.
We have a good question here. I'm interested to see your response to this. Can Nosema be transmitted through pollen bee bread in the hive?
Oh, yeah. As far as I know, Nosema has 2 phases. So we call it an obligate parasite because it does need to be inside of a bee to be considered alive. But it also has what's called a dormant phase. So that dormant phase of Nosema can basically sit and wait inside of, your nectar, your honey, wax even,
Mm-hmm.
and even the pollen and bee bread. So while it might not be alive in there, as soon as it gets consumed and end up in the gut, it automatically just switches back into its environmental phase and isn't dormant anymore. So yes, it can be spread through pollen and bee bread. But with that being said, we do think that there are, different types of pollen, or even like artificial pollen that's actually really good for bees. So again, giving your bees these pollens or special food might actually be good to help them fight nosema infections.
Huh. Yeah, that's good to know. So it doesn't take a lot for these spores to stay alive, right? I mean, they're pretty, I mean, they're like a virus, right?
They're resilient.
Yeah, they can just exist on almost nothing because it isn't like a capsule of DNA. The
Yeah, like basically like a hard shell with like all the DNA inside. So once they, shoot the DNA out, it's
Right.
everywhere pretty much.
Huh.
It's nasty.
So another question is, which honeybee subspecies or race is more resistant to Nosema ceranae, and which one is more susceptible?
Based on my research of what I found at the time, the commercial honeybees are much more resistant to Nosema ceranae versus the Californian genotype, which has more of the African ancestry. And here's why I believe so. It's because these commercial bees have been bred to be able to survive different diseases, such as, Nosema ceranae infections, because beekeepers don't want their bees to die from, Nosema, because we had colony collapse disorder. They know how bad it was for them, so they wanted to be able to have these bees defend from it. But the bees that come from other parts of the world, like the California bee, there's a chance that these bees have not seen or experienced Nosema ceranae before, so they are not bred to be able to defend against those infections. So that's my answer to that.
Yeah.
I
And
think
it
it's
could be that
also,
some of those commercial beekeepers overuse Humidale, right? So the bees have become resistant to that. Is that possible?
Possibly that, but it might also just be their immune systems in general being able to fight against that through different biomarkers.
Yeah, okay. Mad Hat, did you have your hand up? I just caught that out of the corner of my eye. You're on mute, Mad Hat.
I believe some research came from Spain by Raquel Martin, showed that in Nosema cerana spores it can be transmitted through the pollen, and when the bees visit flowers, they can drop the spores, and the spores, it's a form resistant, it can survive. The other bees will pick it up with the pollen, bring it back to the hive, and it can be a source of reinfection. So just I would like to add this one.
You know,
If you look at Raquel Martin research from Spain. Yeah,
Thank you.
you're welcome.
all right. So here's a question is, how do you decide the titer concentration of oxalic acid, et cetera, vaccine that you tested for treatment? And is there an opportunity for lower concentrations of that titer and lower mortality rates if deemed effective?
yeah. So for my experiments, we used the concentration that was naturally found in the seminal fluid. So it's a really tiny amount actually. But I didn't really have a chance to actually play around with doses, right? So it is very well possible that a lower dose would be equally as effective. Um, and again, because I had such a short amount of time to actually do these experiments, there's a lot of questions that we need to still answer before, I recommend using them in your colonies, right? Like we don't know how long they actually, survive in the environment when you put them out, being exposed to heat, right? So does the oxalic acid degrade over time? These things that we don't know, how often you need to treat. So yeah, lots of questions still need to be answered before you decide that oxalic acid is the treatment for your nosema. But there have been other studies that have shown that when beekeepers use oxalic acid to treat varroa mite, they do see a decrease in the amount of nosema spores in their colonies. So, I mean, I don't really have, again, the recommendations, but if you are already using oxalic acid in your colonies, then you might not need to change your routine up at all.
Yeah.
Kind of to bounce off of Jess Webb's answer, I think it's always very important to do your own research using peer-reviewed publications from entomology journals on honeybee topics to make your own decisions of how you treat your bees, because I have experienced many beekeepers who base their treatments off of what they see on Facebook, and sometimes that might not be the right way to go. So always do your own research and make your evaluations based on scientific basis. That's what I think.
And also for evidence-based research that's in the area where you live, right?
Exactly.
Yeah.
So, yeah, okay. So the presentation cited a 90% mortality rate for bees with Nosema. Is that a laboratory phenomena? That mortality rates be the same for any, would that mortality rate be the same for any bee colony?
I don't think you would see no, you wouldn't see as much in a colony, right? So for our cage experiments, it's super artificial, unnatural. The bees are not happy in those cages for the most part. So again, after 18 days, all the bees would die regardless, right? So we try to keep these experiments relatively short, so that, you know, them simply being in the cage isn't a factor that we're seeing when we're treating them. And again, we only put like 30 bees in this cage, no queen, no larvae. So super, super artificial. I wouldn't expect that to actually translate into the mortality that we would see at the colony level. And again, we did, you know, measure the amount of population and the amount of capped brood and larvae, and we didn't see any significant difference between treatments. So I wouldn't expect to see that.
So the same tag to that same question is, I heard somewhere that adding bleach to sugar syrup in fall decreases winter Nosema counts. Is there something in bleach that would mimic the treatment substances?
No, bleach is, you know, an
Fungal.
antimicrobial, or yeah, antifungal, antimicrobial. So it certainly would kill the nosema in your colonies. Uh, Again, do I recommend that? No, because it very well could also kill your bees. It could also end up in your honey, contaminating your frames. Like there's a lot that we don't know about using bleach in colonies. So, but yeah, it certainly would kill the nosema spores in theory.
Well, would it kill the Nosema spores that are in the gut of the bee? I mean, it would kill the spores that are in the sugar syrup, but the bees digest it, then they run it through their, you know, their system. And that, I don't know that you can answer that, right? I mean,
Yeah, I
but
don't think I can answer that either. It would like work as a surfactant almost, right? So if you have the bleach in the sugar syrup and they store it inside of the frames, then it would also, you know, again, in theory, kill the spores inside of the frames. But inside of the bee, I could not answer that.
yeah.
I mean, I'd be so more worried about the bees eating the bleach and it possibly hurting the bees. I mean, we shouldn't drink bleach, so I wouldn't really want bees to also drink bleach.
Yeah.
Well, you know, I had one time in my master beekeeping class, Dale Hill, who wrote in the Hive and the Honeybee. He has a section on nutrition in there. And his recommendation is that for every 5-gallon bucket of syrup that you put out for your bees to forage on, to put a half a teaspoon of bleach in it. And he said that the reason is that it's an attractant. You know, that's why you find bees around your hot tub and your swimming pools a lot. And it also kills any of the, you know, it's an antimicrobial and antifungal thing. And so I do that. You know, I put a few drops of bleach in a 5-gallon bucket. And I know one thing is that my syrup doesn't start showing growth of stuff in it at all. And the bees love it, but who knows
I
what
would
that
also
is.
say the same with people using like lemon juice in their sugar syrup.
That's
Yeah, we
probably
could also kind of validate what we did when we use citric acid, because that's pretty much
right,
all that
right.
eliminates, right? So
Yeah. If the bees were not able to poop or defecate for the duration of your experiment, how do you think this might impact your results?
Well, I would think that if the bees were able to poop outside the hive, since Nosema does exist in in the hindgut of the bee as well. I'm sure that some of the Nosema would leave the bee as the bee's poop, but I'm sure they'd also be able to reinfect themselves with said poop inside the hive and, continue to reinfect themselves. That might change the numbers a bit. So, who knows? I could redo the entire thing and bees be able to be inside of a flight cage instead so they can poop as much as they wanted. But that would be, I'm sure it would change the numbers a lot. And I'm sure
Yeah.
it would cause less explosions when bees, are having their hindguts and midguts removed. I'm sure it would decrease the amount of explosions I experienced too. So
Yeah, that just interesting thought. Let's see, I've had good luck with complete, they said, and that's in quotations, that has Nosema in it and is available in the US. So
Yeah.
just
Nose of
somebody
it's
saying,
interesting
they-
because again, it's like nose of it is like marketed as almost like a bee laxative, right? It's a intestinal cleanser. So if there's nothing in the intestines and they're pooping everything out, I imagine there's no nosema in there at all either. So
yeah, that might change the natural flora.
it's like a good magnesium citrate laxative for us.
Yeah.
Right, yeah. You have
Nothing
to
in there.
rebuild your gut flora, something like that. Well, guys, that's, let's see, we have another raised hand. Let me see here. So
Smith here.
Is
Yeah,
that
just
you?
I'd like to add one thing. I'm, by the way, from Canada, and we did lots of research in Nosema too. Um, the only product actually as registered for Nosema control by FDA as well as Health Canada is Fumagillin. The other products which in the market, basically they try to go around like what you see in the pharmacy, big shelf full of stuff. It can make you sometimes happy or laxative or whatever. It is not controlled and it's not done properly to claim it is a controlled substance. It can have a cause and effect. If you take it, it will affect such. So that's the kind of, I have some guys using Nazavit. I have some guys using bleach. I have some guys add any different things. And being around the industry for the last almost 40 years, some guys use Nazavit for quite some time, and later on they get big kill of Nosema, their bees because of Nosema. And still they believe the bees were died from something else. So this kind of would try to stick here to what we know. In Canada, definitely, especially Western Canada, we again, based on the Nosema spore level, we recommend for them to use a Fumagillin as a certified medication, but it has to be used properly. One thing just to notice, Fumagillin is sensitive to the light. So once you put it in that jar and exposed to the light, there is no use because it's sugar syrup.
Right.
So it has to be done actually with protected from the light, and that way the bees will get it. A couple of things happen here, and we change a little bit the recommendation. We feed the bees with that pail to protect it from the light, 2.5 gallons. If you have weak colony, will never take the dose. So that's underdosing. And later on, if you have a strong hive, which is healthy, and she doesn't need Fumagillin, but it takes all the Fumagillin in. And most of the food will go and stored in the honey combs. And later on, we claim the medication didn't work. Research came and say, if you actually drench the Fumagillin in sugar syrup on the bees themselves, that forces them to take the medication in their body, and that will provide the proper dose to kill the spores, to kill the vegetative form of Nosema. Sorry. So now, actually, we are highly recommending to the beekeepers, if you are going to use fumagillin, you actually, we simplify the whole recommendation. 1, 2, 3. 1 cup of sugar syrup, 2 grams of Nosema, 3 application, once every 7 days. And why 7 days? Because again, there's the spores will come out from the guts tissue into the lumen of the guts and start reinvading again the guts cells. So once we do it 3 times, actually it does help to reduce all of the symptoms. And that's really our recommendation, been going for some time, and it's working for the beekeepers. So yeah, I can understand, but it's a good kind of research you did here. And I'm quite interested because I am from Christine Ping Lab.
Okay.
Yeah, I got my PhD from Davis, and I am from Christine Ping Lab. Okay, so that's she was always in the top of this kind of work. All right, thank you.
No, thank you.
Man, that was great. And we just had somebody pop up and ask, what was the recipe again for using that? Use 3, 2, 1.
I charge $10 Americans.
Go put it on our bill. Yeah.
All right. Yeah. 1, 2, 3. Just 1, 2, 3.
Yeah.
1 cup of sugar syrup, 2 grams of fumagillin, 3 applications. If you add all of this up, 6 days.
Huh.
Okay.
That is
And
good.
actually, the fumagillin now, fumadill is no longer produced by Man Lake because there was issue in the dissolving in the sugar syrup. So now the fumagillin coming back again from Canada in the United States. Because our formulation, actually, it was made and modified to make sure it dissolves in sugar syrup. So that's being close to the industry. So that's been happening now. So if you, just ask your supplier and say you would like to have fumagillin, and I know the company got permission already from FDA to supply the product back to the United States from Canada.
Cool.
Yeah. Awesome.
Yeah.
Okay. What do you suggest for what is the best way to control or reduce Nosema infection in honeybee colonies if Nosema medication is not available? So let's say you don't have access to Fumadill. What would you do?
I think the product is available. Like, frankly, we had one issue here in Canada around 2019. 2018, 2019. That picture actually Dr. Webb showed about fumagillin with a yellow label. That's the old label. Because the guy who used to own this company actually passed away, and later on we had disruption of production for 1 year. And actually at the time, this is a long story, but at the time I worked with the Vita Health from Europe to take over the product, and they produce it now in Canada, and it's been in the market, and we never had any short supply of that because, frankly, our bees in Western Canada, we are reliant almost 100% in fumagillin to get the bees through 5 months of winter nidification. And I would love to send the bees to California to stay alive during winter time, but we really rely on it. So, frankly, yeah, it's available. The only problem, sometimes some beekeepers think it's too expensive. But if it costs me $5 to treat the hive, guess what? I save a colony for $200 or $300.
Yeah.
And that has to be considered.
Right. Right.
Yeah.
All right. So this
Thank
one.
you.
Yeah. Thank you. So I think, Dr. Webb, you had talked about citric acid instead of bleach in syrup. And somebody asked, what's the ratio? How much do you put in, per say per quart or
Yeah.
gallon?
Again, I'm not recommending that you use this until, further experiments. But if I remember correctly, it was something super small, like 200 milligrams per like a 32 fluid ounce mason jar. So it's not a huge amount of citric acid.
Yeah, and it's non-harmful, so I mean, it's not like you're going to
Yeah.
OD them on lemon juice, right?
Right, right.
So, okay. So what are the risks of Nosema infection associated with the movement of commercial bees to California for almond pollination?
Well, I mean, there's a paper that I read, Trainer et al. 2016, and they did find that bees that moved from, place to place migratory beekeeping, they did have higher levels of Nosema infection than bees that were stationary. So I'm sure as bees get exposed to different temperatures, different, lots of different things as they're traveling, perhaps, they have increases of infection based on that alone.
And it could be, as you said, Dr. Webb, a comorbidity, right? So they have a high, say, mite infection, and then they're more susceptible to manifesting Nosema.
Yeah, I think migration is super specific because one, they're trapped in their colonies for so long, they can't actually leave to go defecate, which means they're, probably defecating in the colonies or either holding them in for a super long amount of time. Which means if they are defecating in the colony, then it's spreading in the colony, right? We know
Right.
bees are normally pretty clean and they try to leave to poop if they can. So this creates like a microcosm, like a really good place for Nosema to grow. And then we also know that when they're out in the almonds, they're only getting one type of feed. And we, again, already talked about how nutrition is going to be really
Yeah.
important in
Yeah.
helping them fight off the Nosema infection. So only having almond nectar isn't necessarily good for them either. So it creates a really special place that Nosema really likes.
Yeah. Yeah, that makes sense. There's a comment here in the chat that says citric acid, that there was a study in Norway that said a 1% sugar syrup solution is a purgative, it looks like, for, as a possibility. So,
Yeah. Again, really small amounts should.
yeah, not much.
Yeah.
All right, sorry keeping you guys so late, but this is in bee language, you kicked the hornet's nest here. So there's a lot of questions coming in. I think that's about it. Well, again, thank you all, and to the presenters for the work you did to get ready for this. It was very, very interesting, and so we look forward to maybe hearing from you down the road in the future. Did you guys learn anything as well from us?
Oh yeah, for
A
sure.
ton.
I always love talking to beekeepers.
Yeah.
Yes.
Well, Medhead, thanks for jumping in and helping us too. You're always a wealth of knowledge and
Thank you.
always a welcome addition to our mini conferences. So the next, so you can look at this on our website. It'll be posted here in a few days, and our next mini conference next month will be on rare honeys, and we have a beekeeper who on our board actually who deals in rare honeys, and so he's going to talk about that. In December, we're going to take a pause because of the holidays, but we're going to post some stuff on our Facebook page, the honey recipes for the holidays that you could use for baking or whatever. And then we begin under our new website and new virtual platform in January with Dr. David Peck, who's going to present to us in that one. So thanks again to everyone, and join WAS. We can always use the membership. That's how we sponsor these sort of things. So it's the best $35 you'll spend having these opportunities every month. Thank you. Good night to everyone.